Today we're going to talk about the subject of genetics, and we'll first start this lecture talking about a very interesting fellow by the name of Gregor Mendel, as he became known, born Johann Mendel, but became Gregor Mendel. And essentially he was a farmer. His family was from a farming background and had a lot of experience in farming. His family had owned this particular farm for over 130 years in his family line, and so he started off early in his life with a lot of experience in the farming world, growing food and things like that. And he studied. He was quite good at science, and he studied physics and math and astronomy and meteorology, and became quite a well-educated person over the years, and also studied theology and was quite religious. And so he eventually ended up working at this monastery and became an Augustinian monk. And in his sort of role, he also was teaching high school, but in order to teach high school he had to pass a series of exams, which he took on a couple of occasions, but was not able to pass. He always had trouble passing the oral section of this exam, and so essentially he had difficulty and was sent back to study more physics and math. And during this time he would get quite ill. He suffered from bouts of depression, and we believe maybe kidney infections as well. And so he had a kind of a hard life. And in the meantime, in his studies of working as a monk and having this really well-established background in physics and math and in botany, plus his farming experience, sort of all led him on the side to perform these pea plant experiments. And so in 1863 he published a paper basically on the hybridization experiments of plants. And if you remember, Charles Darwin's theory of evolution, which was first written up in about 1844, was published in 1859. So you notice that they're working together at about the same time. I say working together, they didn't actually know one another, and necessarily did not know about each other's research, but they were doing the same, doing their research at about the same period of time. So one of the interesting things is that when Mendel did his work, it was largely unnoticed, and it was just thought of as sort of a hybridization and not really known for the genetics side of it that later, about 100 years later, became apparent. So it wasn't until about 100 years after his research and well after his death that we sort of rediscovered Mendel and everything he had done, and then tied that into Charles Darwin, who was also had passed away at that time. Sort of were able to blend the idea of natural selection and evolution with modern day genetics, which did not happen during Darwin's time or Mendel's time. So this is Gregor Mendel. This is his garden right here. It's about 5 acres. It's about a quarter of the size of our wildlife sanctuary. So had a kind of a good size area, but the kind of area that you have to pick the right organism to study. And so what Mendel worked on, among other things, animals and such, the thing he really became famous for and could really do a lot of experiments on were on these pea plants. And so the beauty of pea plants, like many plants, one is you can grow them pretty quick. You can have a whole bunch of them in a small area. We think by the time Mendel was done, he grew something like 29,000 different pea plants. And so one of the things with pea plants is they have on them, like many flowering plants, they have both male and female parts on them. So they have male parts and they have the female parts of the flower. And so what you could do is plants usually will not self-pollinate, meaning, there's usually some mechanism in place so that this flower won't pollinate itself, but you can kind of control that. You can choose this flower to mate with another. I'll show you the next picture. And so what happens is you have this pea plant, and the sperm or the pollen will pollinate and then fertilize the eggs inside the ovaries here, and then what you end up with is the seeds, and then from the seed this thing grows into a new pea plant. So that's what we call like the life cycle of a pea plant. It's going from the adult with the male and female parts. The male parts go through meiosis to produce the pollen, which contains sperm, okay, and then the female parts produce the from the ovary will produce the egg. So there's my egg, and then the sperm fertilizes the egg, and that gives me a diploid cell, a 2N cell with 2 sets of chromosomes in it, and that it eventually can become that plus the other cells surrounding it can become the seed, and that can grow into a new pea plant. So the beauty of working with something like pea plants rather than mountain lions, which would be very hard to control, is what you can do with like a paintbrush is you can go in and you can say, okay, I want to take this flower and I want to mate it with this flower. So you can go in, you can cut off the male parts of one flower, and you can go over here with a paintbrush and you can pick up some pollen from there, and then you can pollinate this flower. So instead of having an insect do this like a bee, which Mendel also was known for his studies on bees and growing had beehives and things like that, is that you could take this paintbrush, you could swab it over here like that, pick up some pollen, and then transfer it over here and fertilize this particular plant. So this plant, you could control its breeding with this one. We cut away the male parts often just so that this doesn't accidentally fertilize it. And so we know then that this plant has now pollinated this plant. And what he found was that there were certain traits he worked with. We'll get into the second here a little bit later about maybe how it's a little tricky how he came up with these numbers. But he found, for example, if you take a plant with yellow seeds, all right, and you take a plant with green seeds, that when you breed them together, what you would get, and we would call this the P generation, either the parent generation, or you'll also see it referred to as the pure breeding generation. But these yellow-seeded plants, if you cross them with themselves, if you breed them with themselves over and over again, what you would find is you always would get yellow-seeded plants. This also is in the P generation, the green-seeded plants. If you were to breed these over and over again, you would always get green-seeded plants. So these always gave you yellow, these always gave you green. But then what happens if I take a yellow-seeded plant and I cross it with a green-seeded plant? So that's what they did here. And what they found, or what Mendel found, was that in the F1 generation, which stands for filial, which is basically the children or the offspring. So the filial generation, the F1 being the first children generation from the P generation. What you would find if you mixed yellow flowers with green flowers is what you would get, yellow seeded plants with green seeded plants. What you would get in your F1 is you would get all yellow seeded plants. So 100% would be yellow seeded plants. But then Mendel would take this and cross it and find that the green information was still in there. Up until this point, most geneticists of the time in the 1800s and all the way up into the early 1900s, our view of genetics was that the information from both parents was sort of mixed, meaning that you would get, if you did this kind of thing with most genetic information, that the information with the yellow, whatever made them yellow, get to that, and whatever made these green, gave you sort of a mix, gave you sort of a yellowish green. And Mendel was the first to show, well, that's not always true. There's that you can get these where the information, where only one set of the information is passed on, although the other set is there. All right, so it's a good time to introduce some genetic terms that we need to discuss. These are things that are sort of added to Mendel's work. He didn't really use many of these terms, but we use them now, so I'll put them in here. And some of them he actually did use. First of all, we have what we call the phenotype. The phenotype is the physical outward appearance of the genetics. So, for example, with our pea plant, the information that made them a yellow-seeded plant, the phenotype of that would be that they're yellow-seeded or green-seeded. So the phenotype is the expression of what the genotype does. So the genotype then is the actual genetic makeup. So we often use letters to represent that. So for example, with the yellow-seeded plants, we'll use a big Y to represent the yellow. Okay, and then it turns out the green is that same information, but it's slightly different. So instead of using a G, we use a little y, and I'll explain why that is later on. But anyway, that represents green. So the genotype is the genes. It's what the actual genetic information is that gives you the phenotype. The phenotype's what it looks like, or the expression of the genes. The genotype is what the genetic makeup is. Probably already mentioned this already when we were talking about transcription and translation, but the idea of a gene. So you know what a gene is somewhat just from your general knowledge. A gene is something that is part of your genetics. It affects the way you look. But specifically at a molecular level, a gene is a piece of DNA or a section of DNA that is responsible for making a protein, or at least part of a protein. So remember that living things are largely controlled by their enzymes. So the enzymes control all the different stuff that we can do. Making your eyes blue or brown, and making your skin light or dark, or making you tall or making you not so tall. A lot of that has to do, not all of it, but a lot of that has to do with certain genes that you have. And then those genes interact with your environment and give you certain features. Well, the gene is basically a protein, and basically, for the most part, the protein is an enzyme, and the way we control, or the way we look different from one another, is the simple fact that we have different genes, which therefore make slightly different enzymes, and those enzymes give you slightly different chemical reactions going on in your body that create a variation. Okay? Now, when we talk about variation, it turns out that gene, a lot of genes come in different forms. So we call that an allele. An allele is an alternate form or a different form of a particular gene. So, for example, there are genes, and we're going to make this a little more simplistic than it really is, but let's just say for a minute that there are genes that make your eyes blue, and there are genes that make your eyes brown. And actually, the gene, the location of that information, is in the same spot. So we have different genetic codes that relate to whether you're going to make a gene that makes your eyes blue or makes them brown. And again, I'm making this quite simplistic. It turns out there's hundreds of genes that affect your eye color, but there is sort of a dominant recessive thing going on where there are certain genes that have a large impact on your eyes being brown versus non-brown or blue and non-blue. So there's some other genes that affect that, but there is certainly a sort of a general statement you can say that that's pretty much true. And it turns out a lot of times when you have these alleles, one of the alleles will be dominant and one will be recessive, one or more. Dominant simply means that if you have that allele present, if you have that piece of information, then you will see the effects in the phenotype of that with just one copy. Okay, if you have one copy of a dominant allele, you'll see the effects of it. Recessive means that you need to have that be the only thing present, meaning that if you have a dominant and you have a recessive and they work and they're the same gene, So the dominant gene or the recessive gene, if you have one of each, you won't see the recessive one because it is overpowered or hidden by the effects of the dominant gene. Okay, so we'll explain that more in a second here. All right. Other thing we can talk about is some more genetic terms, this idea of homozygous and heterozygous. This simply means, homozygous means that you have two alleles that are the same. So here we have P, and little p, for example. That's homozygous. Homozygous because they're both the same. Heterozygous means you have two alleles and they're different from one another. So big P, little p. So homozygous, heterozygous. Homozygous means the two alleles are the same. Heterozygous means they are different from one another. And the other thing that's important is this idea of cells being either haploid or diploid. We mentioned this also in mitosis and meiosis, but remember that haploid cells have one set of chromosomes in them. We use the letter N often to represent haploid cells. And diploid cells have two sets of chromosomes, and therefore that's 2N. Remember also that the number in a set varies by organism. So in the case of humans, for example, the number in a set is 23. So the number in two sets then is 46. So humans have 46 chromosomes in them unless there's some kind of error. And in haploid cells such as sperm and egg, there's one set and it's 23. But it varies. You can't really tell how many chromosomes there are in a set unless you know how many that species is supposed to have. So I just want to make a point that if I were to ask you, okay, how many chromosomes are in one set, you have to know what the animal or plant is that you're dealing with. So if it's a human, it's 23, but if it's a horse, it might be 25. If it's a fly, it might be 2. It can vary depending on your species. Okay? Now, these are some of the traits that Mendel worked with and whether they were dominant or recessive. So you notice we have seed shape, for example. They could be smooth or wrinkled. And it turns out that the dominant trait was smooth and the wrinkled trait was recessive. Or they could be yellow or green, for example, or inflated or wrinkled, or green and yellow, or the flower color. You could have purple flowers, or you could have, white. You could have white flowers. So purple flowers or white flowers. And one was dominant and the other was recessive. And what's interesting about it is that these are the primary traits that Mendel worked with, but looking back now, over 100 years later, we started to replicate these experiments. It turns out there's all kinds of traits that pea plants have, and the ones Mendel did his study on here, all were kind of nicely dominant and recessive. And as we'll learn later on, they were on different chromosomes for the most part, or very, very far apart on any chromosome. So his numbers worked out surprisingly well. And so there's some debate whether Mendel, it would be extremely lucky for him to have done these experiments on these 7 traits and only these 7 traits and got this data, because it turns out if you look at some of the other traits, the numbers don't work quite so well. We can explain that now and why that is, but during Mendel's time, we believe maybe that those parts of the numbers did not work out right. So either he got extremely lucky, or perhaps some of the data he threw out that he was not able to explain, and we'll talk about that later on. What I want to do now, I want to talk about, remember at the time, so structure of DNA was published on the structure of it in 1953. So this is about 100 years before our understanding of the structure of DNA. So in the 1950s, we understood DNA. We started to understand the role that DNA played in inheritance and things like that. But up until that point, this 100 years ago, so there's really no knowledge of DNA and what the DNA molecule can do and so forth. So what's interesting is we can put that all together now and kind of show you. So we have this pea plant here, and let's say we're talking about the yellow seeds. Okay, how does this plant have yellow seeds versus green. Well, if you take this plant, anywhere in the plant, take the plant and you look into the plant, you find the yellow seeds. And if you look at a microscopic level, you would find that the plant has cells. Okay, so that's no big surprise. We've talked about that already. So here's our typical plant cell found through this whole plant. And if you go into the plant cell and you go into the nucleus of the plant cell, there's a nucleus, and inside the nucleus is the DNA. The DNA has a certain code to it, A, G, T, C, and so forth, all the way through. And there are certain sections that you read called genes. And you might start here, and you might travel along like this, and you might copy through transcription into RNA this piece right here. So that becomes then with some editing our mRNA comes out, and then it's read by the ribosome. And remember, the ribosome is used to sort of hook up the mRNA with the little tRNAs, and that then gives you, when you read this in a codon sequence of 3, it gives you a section of amino acids when you're done. So there's my amino acids, and those then, that chain then will fold up, and perhaps with another strip of protein, or perhaps just that one, will fold up into a 3-dimensional shape, and that will give you your functional protein. If that protein works as an enzyme, that one of the things that can happen is the way you have these yellow seeds is this protein is able to take the molecules in those seeds and convert them or break them down into some other product, and those products become yellow pigments or green pigments. Now in this particular drawing here, I just picked an enzyme, so this one shows you the breakdown of sucrose here. So this is actually the sucrase enzyme making glucose and fructose. So I'm not actually showing you this particular enzyme, but the idea still holds that you have a protein of some sort that can alter the shape of the molecules in here. And if they're a pigment molecule, probably the standard colored pigment is a green color. That green color gets turned into a sort of yellowish color, and that gives you your yellow seeded plant. All right, and one of the things that Mendel did is kind of interesting is we would always get this 3-to-1 ratio. We'll talk about in a second, this 3-to-1 ratio, 3-to-1. And it's important to note that Mendel, because he was so good at math, you just didn't get 3 and 1, which would make it easy. He would get like 787 to 277 and 5,474 to 1,850. So he was able to sort of break those numbers down and realize they were a 3-to-1 ratio. And here's a picture here showing you sort of some of his pea plants here. You can see the wrinkled seeds and the green seeds and the yellow smooth seeds in there. And so what he essentially did was he said this. He said, what we can do is we can take the plant and we can look at it, and what we do now is we draw these things called Punnett squares. So the Punnett square is designed to show you the chances of certain events happening. Okay, so if you have the big P and the little p, and the big P and the little p, and you cross those, so this is one plant, this is another, what you get is you get this plant right here, big P, little p, and since that has one dominant trait, that flower will be purple. Okay, and then over here, this one has a little p and a little p, and that gives you white. Okay, I should go back here and point this out for a second, just really quickly here. When we talk about the big P or the little p, what that represents, the big P is designed to represent a piece of DNA that can make an mRNA that when you code for it and make your protein will make your flower purple. Okay, so the big P is designed to represent a gene or an allele that when you code for it, it makes a protein that makes the flower purple. In that same location, in the same piece of DNA, on a different chromosome, same kind of chromosome, so chromosome 1 and on the other chromosome 1, for example, because you got 2 of each, 1 from mom, 1 from dad. If that sequence is different, okay, then we use a little p, a lowercase p, to represent that same piece of DNA, but it has a different code to it, which instead will give us, I guess, purple, it was white flowers, right? Okay, so in that case, white was recessive, so the lowercase p. So we use the p normally for the dominant trait, and then the lowercase p lets you know, instead of using W, which would imply it's a different gene altogether, the lowercase p lets you know it's the same gene, but it's a different sequence. It's a different allele in that same region, okay? All right, so once again, if we look up here, just to show you sort of how some of these Punnett squares work, here I'm taking a yellow plant that is heterozygous because it's got but one big Y and one little y, one big Y, one little y. If I cross those, you notice I get yellow, yellow, and green. So here's a yellow here. That's the phenotype, it's yellow, but the genotype is big Y, big Y. Over here, this one is yellow also, so these look the same. But genotype-wise, if you look at the genotype, this is big Y, little y. Okay, big Y, little y. So different genotype. This is different in both the genotype and the phenotype because this one's green because it has 2 copies of the recessive. All right, this just shows you sort of how they are, where they're located on a chromosome. So the idea is there's chromosomes, and in those chromosomes, there's sections of DNA that can code for the certain genes, and you have one set of chromosomes from each parent. So mom gives you one set and dad gives you one set. So the idea is your chromosome 1, you have a chromosome 1 from your mom and you have a chromosome 1 from your dad, and that information could be the same in that particular spot, which could give you yellow seeds, for example. But your mom could have the dominant trait, your father could have the recessive trait, and you still get, because you have one copy of the dominant trait, you get the heterozygous version of that, you still have yellow seeds. Or both parents could be green. Green would give you always green because you have homozygous recessive, you have 2 recessive copies, that gives you green as well. Okay, so let's sort of put that together now and look at this figure here. So here's what kind of one of the things Mendel did. So we had a female flower and we had a male flower. And the female flower, the P generation, gave me always yellow seeds. Okay, so it was a pure breeding or parent generation, P generation, big Y, big Y, big Y, big Y all the time. Now Mendel didn't know. He knew there was information there, but again, he didn't know about the genetic side. He didn't know about the DNA structure. He just knew there was information there that could be passed on. And before this time, remember, we thought if you mix the Y and the green, you got a sort of a intermediate color, which sometimes you do, and we'll talk about that later on. But Mendel was able to establish just sort of basic rules of how this works. So what happens is, the female plant and the male plant, they both go through meiosis to make haploid cells. Okay, remember, sperm or egg. Because if they don't do that, remember, they pass on 2 sets, and instead of having a cell that had 2 sets of chromosomes, if they don't go through meiosis, you end up with 4 sets of chromosomes. So we go through meiosis to take the information, we copy everything in it, we divide it twice, and we end up with gametes, and gametes are sex cells. And the idea of the Punnett square is it's designed to show you all the different opportunities, all the chances that could happen. So in this particular case, the only kind of sex cells this could make is a big Y and a big Y. So that's what you see here. You could make a big Y, in this case the female egg. Doesn't matter which side you put these on, just as long as you have all the options there. So we have big Y and we have big Y. And over here we have little y and we have little y, because those are the kind of sperm you can make. So then what you do is you say, okay, what if this sperm here, or this pollen, which has the sperm in it, fertilize that egg? You would get that, big Y, little y. What if this sperm or pollen fertilized this egg? You would get that, Okay, and so you notice if you do this in the F1, as we mentioned before, in the F1 you get all yellow seeded plants. All right, so phenotypically they're the same as this one. They're yellow. But if you look at their genotype, they're not the same. Okay, the genotype is they are heterozygous because they have one copy of each. All right, so then what we're going to do is we're going to take my plant we just started with. That's my F1. Okay, so here's my F1, big Y, little y. It's heterozygous. All right, and I'm going to pollinate that. I'm going to self-pollinate that. Remember, that's the thing you can do with pea plants is I can go in with a paintbrush and I can fertilize this same flower so it reproduces with itself. And what you found in the F2 then is you would get this 3-to-1 ratio that Mendel kept finding, 3-to-1. You would find 3 yellow plants for every 1 green one. And then what happens is you take those pea plants and you cross them with themselves. You self-breed those. So this is what we call my F2. My F2 gives me my 3-to-1 ratio. Okay, but if I cross that to get my F3, so I'll notice then if I take one of those, and you don't know, these all look yellow and this one looks green. If I take my green one, I'll get green. So that makes sense. Okay, but it turns out of these, a quarter, 1 out of 4 of these, or 1 out of 3 of the yellow ones, if you breed it with itself, you get all yellow. Okay, because it's YY. And over here what happens is these end up giving you that 3-to-1 ratio again. Because if you take these two and cross them with themselves, you get big Y, big Y, big Y, little y, big Y, little y, and little y, meaning these two are the same as these in your F2. Your F2 generation, at least those two ones right there. So these individuals here end up being just like these, and gives you that same result, that 3-to-1. All right. Now, what happens if we take 2 traits at once? What if we take a pea generation that's both smooth and yellow? So smooth is dominant, it turns out, over wrinkled, and yellow is dominant over green. What happens if we take smooth and yellow and cross it with wrinkled and green? What we find is when we cross that in the F1, 100% of them are smooth and yellow. This is called a dihybrid cross. Dihybrid, because I'm crossing 2 different traits. Dihybrid cross. So what happens here is I get smooth and yellow for all of them. Then in my F2, I'm going to cross that, and what I get is I get 9 smooth and yellow. Whoops. I get 9 smooth and yellow. I get 3 wrinkled and yellow. I get 3 green and smooth, and I get 1 that is wrinkled and green. So a 9 to 3 to 3 to 1 ratio. And the way that works is you have to look at it like this. You have to go through once again and make your Punnett square, but you notice it's a big Punnett square. This one has 16 boxes, because remember, the Punnett square is designed to show you all the chances of all the events happening. So it turns out my smooth and my yellow, they're totally separate genes. They're like on different chromosomes, and the smooth does not affect the yellow. So those are independent of one another. And that also goes with green and wrinkled. So what I have to do is I have to go, okay, when this plant here, when it goes through meiosis, it's going to give 1 set of chromosomes. So it can give 1 set with the big S and 1 with the big Y. It's got to do 1 of each because they're on different chromosomes, and you're going to get 1 set. So you can make a gamete or a sex cell that has a big S and a big Y in it, or you can make a gamete that has a big S and a big Y in it. And so no matter how you do it, it turns out the only one you can make is the big S and the big Y. This one, this S could be packaged with that Y. This S could be packaged with that Y. This S could be packaged with that Y. And this S could be packaged with that Y. And so once again, you get the same thing. Small s, small y. So then what we do is we take those, we cross those, and then my F1, just as I said before, no big surprise, I get 100% of them being yellow and smooth. But if I cross that, notice that over here I have my sperm, and over here I have my eggs. And remember, it doesn't matter which side you put them on, just as long as you have all the options. But when this goes through meiosis, remember, it's going to put in one set in each cell. So if this was a human, there would be 23 different chromosomes. But we're only interested in here, if this in our pea plant, we're not interested in all of them. We're just interested in the ones that have these genes on them, because that's all we're paying attention to. So this S here could go into an egg, for example, that has that Y. This S could go into an egg that has that Y. And so that's what you see here. You see big S, big Y, big S, little y, little s, big Y, and little s, little y, because those are all the different options that I could have. Those end up being the same down here for the egg. So the sperm and the egg end up having the same options. And then I just simply say, okay, well, what if that big S and big Y mated with that one? Then what I would end up, if that sperm got with that one, I would end up with this. Big S, big Y, smooth and yellow. Okay, so if you count all those up, you should find 1, 2, 3, 4, 5, 6, 7, 8, 9. So there's 9 that are yellow and smooth. Okay, then I count how many of them have just one of the recessive traits in their phenotype. And I noticed that, okay, for green, there's 1, there's 2, there's 3. So there's my 3. And how many of them are wrinkled but yellow? There's 1, there's 2, there's 3. So there's 3. Okay, how many are green and wrinkled? Notice there's just that 1. Now, that's the Punnett square way to do it. It turns out there's sort of a mathematical way to do this as well. So we introduce this idea of rule of addition and rule of multiplication. And what you can do is you can say, okay, what are the chances of 2 independent events happening? And if you ask yourself, what are the chances of one happening or the other? You can add the chances up individually. It's called the rule of addition. So let's say in this particular case, I wanted to go, well, what are my chances of getting a seed that is either green or wrinkled? Okay, what are the chances of that happening? All right, so rule of addition, if you did each Punnett square, right, if you do the single Punnett square like this, and you do them one by one, and you say, okay, if I cross that with that, only 1/4 of them, 1 out of 4, would be green. So that same thing would apply to wrinkled, and I would also get 1/4, right, if I did them independently. So rule of addition is I would add those together, and I would get 2/4, because remember, your denominator has to be the same, and then you just add the top numbers, the numerator. And so 2/4 then can be reduced down to 1/2. So if I look over here, I have 16 boxes. Okay, and so I have 1, 2, 3, 4 that are green, and I have 1, 2, 3, 4 that are wrinkled. You have to count this one twice because it has both. So that gives me a total of 8 out of 16, which is also 1/2. Whoops. Sorry, 1/2. I said it right, but wrote it wrong. 1/2. So that's the rule of addition. Okay, rule of addition is if you have 2 independent events happening, and you ask what are the chances of one happening or the other, you add the individual chances of them happening to get your answer. Okay, rule of multiplication is if you ask what's the chance of one happening and the other. Okay, so for example, I did my same thing up here. Okay, what if I wanted to know what are my chances of it being green and wrinkled? So what if I wanted it to be green and wrinkled? Okay, so much more difficult if you think about it. Now I'm not asking one or the other. I'm asking for both to happen at the same time. So you do the same thing. You take your 1/4, but this time you multiply it by the chance of the other thing happening. And when you multiply, you just go straight across and multiply the numerator with each other and the denominators with each other, and you get 1/16. So that down there, you see, that's the only one. That's 1 out of 16. That gives you your chance of both of those happening. Those ideas led to Mendel coming up with what he called the law of segregation and the law of independent assortment. And the law of segregation, first of all, simply states that when you have copies of genes, that when you go through gamete formation, which is meiosis, that they separate. So each organism has 2 copies of each gene, all right? And that's because you have a piece of DNA from mom and you have a piece of DNA from dad. And so during gamete formation, these are going to separate. And so if you're, let's say you take your mother, let's say this is your mother and she has 2 chromosomes, this chromosome 1 and this chromosome 1. During meiosis, during gamete formation, when your mom goes to make eggs, they're going to separate, and unless something goes wrong, you're going to get 1 copy. Although, remember, there's crossover, so they can swap pieces with each other. But essentially, when they go through and they make an egg or a sperm, you're only going to get 1 of those copies. That's the law of segregation, that the information separates from one another. And then the law of independent assortment is simply that genes separate independently from one another. And it turns out this is not always true, as we'll see later on, but in basic Mendelian genetics, it turns out to be true, where essentially what happens is, what happens with the yellow gene, for example, if you're talking about yellow versus green, versus what happens with the wrinkled versus smooth, what happens with the yellow is independent of what happens with the round or smooth or wrinkled. Okay, so yellow has no effect. These two are independent of one another. For the most part, we'll see that's not always true. Sometimes when you inherit one gene, you inherit another gene closely related to it on the same chromosome. But if they're on different chromosomes, whether you get the yellow allele or the green allele is independent of whether you get the wrinkled allele or not.